it's a shame that we can't be face to face, but it's amazing that we can use some of these technologies to share the incredible work that we're doing at the John Curtin School of Medical Research in neuroscience. Today, I'm going to be talking about some of the work that our group does, Clear Vision Research, in understanding vision sciences and looking at specifically what happens as we age and how the effects of losing specific cells, especially photoreceptors, happens. So today I've titled this talk, Bring into Focus the Importance of Vision Sciences. And so I'm reminded, similar to the wonderful talk we just heard from Nathan, that looking at history and looking at wonderful people can give us insights into science and things that were attributed with it. And Helen Keller was an incredible author. She was an incredible entrepreneur. And she was also profoundly deaf and had no vision at all through a viral infection, I believe, that she got when she was around 19 months. And so as a consequence of that, though, she was lucky enough and fortunate to have an amazing educator, Anne Sullivan. And Anne Sullivan actually trained her, and they worked out a language. They were able to communicate. And they were able to communicate so well that Helen Keller was able to actually not only just communicate through her language, but she was actually able to speak. And one of the amazing things that she wrote in one of her books was, the only thing worse than being blind is having having sight, but no vision. And I think there's a very important lesson there for everyone there about having a vision, right? But what is vision? Well, our definition of vision is an interaction of receptors in our eyes and the environment, light in this case, which is then perceived by our brains as an image. And I use the word perceived there because the way humans perceive the world is very different to the way other animals can actually perceive the world. And even though we're separated by kilometers because we can't actually be together, I can somewhat, potentially, if this video works, excellent, I can manipulate your vision, not me, your own eyes. I want you to have a look at this image that's now flashing up on the screen. If you stare at the crosshairs there and you look very closely, what you may start to see over time is that the pink dots that are there, one of them starts to turn green or flashes up green. Right? I'll just let this play for a little bit. Now, there is no green dot. What is happening is your brain is perceiving the movement. It is then saying, because that movement's there, something must be put in place, and it actually replaces that with a green image. Now, over time, what will happen is if you look at that, the pink dots will start to disappear, and the only thing that will be left will actually be the green dot, if you're lucky enough to see that. And what's happening there is some of your photoreceptors, the cells that are responsible for taking light and converting it to an electrical signal of your brain, become saturated, and it's your perception of that vision is being warped a little bit. And we can do that in another way. So if you look at this particular video, you'll see that this gentleman here on the far left is significantly taller than this individual down here. And if you look at these individuals in the back, they are organized in height. But as I play this video, you'll notice that these individuals are essentially, the perception of their height is being skewed because the way the room is designed is to skew your perception to be able to determine depth. And this is referred to as an Ames room. And once we're able to travel and get out again, I highly recommend you go and see Questacon and look at the Ames room in there, and you can see this visual perception for your own. So vision is a perception, but how do we perceive things, and how is it that we're able to see the world around us? Well, here we have a schematic view of your eye, and light enters into the front part of the eye. It passes the anterior chamber and enters into this part of the eye that will focus the predominant of the talk on, called the retina. And when light hits this retina, it would be coming from this location at the top here. It passes through all of these cells until it hits the outer nuclear layer, or the photoreceptors. And these photoreceptors work with the retinal pigmented epithelium to take light, convert it to an electrical signal that is then sent through the inner nuclear layer through bipolar cells to ganglion cells that form a bundle called the optic nerve that's sort of like a PowerPoint, plugs into your brain. Now, as John said at the start of the day, the eye is a part of the central nervous system and is part of the brain. So that's how it's all connected together. Here's a bit more of a schematic diagram, bringing all of this information together. And what I want us to focus on are these photoreceptors here that are referred to as either rods and cones, and the retinal pigmented epithelium that underlines them. And it's these two cells that work together, these neurons and these epithelial cells, that enable us to take light and convert it to an electrical signal that our brain can then interpret as vision. So what are rods and cones? Well, there are types of photoreceptors, and rods make up the majority of photoreceptors in our eyes. There are approximately 120 million rods in your eyes, right? All working now. There are about 6 million cones, and it's really the cones that provide you with your ability to read, write, drive, and daily interact. They provide you what we refer to as high visual acuity, the ability to see and discriminate small pixels of information, right? And they provide you with color vision. And they are jam-packed into a part of the eye that we'll talk a little bit about later, called the macula, right? So, but I wanted to talk a little bit about neuroscience and how this actually works. And so expanding on some of the great stuff that you heard from Brian, right? I wanted to show you what the actual process was of turning an environmental stimulus of light into a chemical stimulus in photoreceptors that leads to a synaptic response across these amazing neurons. So in a rod, there is a process called phototransduction. And this looks like a really complex diagram, but it really isn't. We're going to break it down now. So in phototransduction, in the dark, let's start off with, what happens is there is a channel that causes an influx of sodium and calcium that hyperpolarizes the cell, which we all learned about from Brian today. And that process leads to the release of a neurotransmitter called glutamate. So actually in the dark, your eyes are producing the neurotransmitter, but it's sending it to cells and it's actually inhibiting them from functioning. So it's an inhibition reaction that happens. Then when light is turned on, it hits a specific protein called rhodopsin. Now this occurs in rods. In cones, there are different sorts of opsins, but we're going to focus just what happens in the rods for today. And when that light hits rhodopsin, what you get is this chemical cascade that leads to the change from cyclic GMP that keeps the channel open to GMP that is no longer able to bind to that channel, stopping the sodium and calcium from actually being able to enter in, and inhibiting glutamate that then allows those second order neurons, those bipolars, to actually fire. So a really simple question. What would happen if your photoreceptors were no longer present or no longer able to work? Well, very clearly, you would be blind, right? This is referred to as vision loss and is caused by most retinal degenerations. So what is a retinal degeneration? Well, it's a loss of some of your retinal cells, and these could be the ganglion cells, these could be your bipolar cells. We focus on the photoreceptors. Now, if I was to peer into the wonderful universe that is your eyes, and we went to an ophthalmologist or an optometrist and they looked in, what they would see in the back of your eyes is hopefully something that looks like this. This is a fundus image of the back of your eye. You can see the optic nerve head over here with the beautiful vasculature coming through. And there's a region right in here. It's only 5.5 millimeters, 5.5 millimeters in diameter. And this provides you with 95% of all of your usable vision. It's referred to as the macula. Now, this is jam-packed full of those cone photoreceptors that, remember, cones give you your high visual acuity and all of your color vision, right? And a sheer, just 1.5 millimeter lesion in that area renders you legally blind. So look at all of this real estate that exists. That is full of other photoreceptors, but it's really this vital region of this macula, this packing in of photoreceptors that, if lost gives you a loss of vision. So 1 in 7 Australians will experience some form of macular degeneration in their lifetime. This costs the Australian economy $5 billion annually. And for the version of this disease that we work on, there are no treatments and cures. And this is the version that we work on. We work on dry age-related macular degeneration, or atrophic AMD. It is the #1 cause of blindness in the Western world. 2 million people globally are affected by this disease, and there are no treatments for dry AMD at all. We focus on oxidative stress, and I believe there was a question before about microglia, and I'll show a little bit of a diagram in a second about microglia and how they're involved in this disease. But the inflammatory cells in the retina are mostly perpetuated by, the inflammatory cells in the retina are in fact microglia, and these contribute to AMD. So what would our vision look like if we had age-related macular degeneration? Well, here's a normal fundus, that's what it looks like, and here's hopefully a vision that you'll all be able to experience one day. Age-related macular degeneration, the dry form, you would have this central loss of your vision, and here you can see some degeneration happening to the macula. There is another form, which is about 10% of all AMD, called wet AMD. There are treatments for this. This is where you have to have a monthly injection of an anti-vascular endothelial growth factor to stop. Essentially what happens is you get a breakdown of the blood-retinal barrier. You need to stop that, those vasculature growth from occurring. So if you inhibit VEGF, the vascular endothelial growth factor, you can stop these bleeds from happening. This is a very acute form of the disease, and it happens very, very rapidly. There are treatments for these. They are somewhat effective, but there are no treatments for the more prevalent dry age-related macular degeneration. So what is the mechanism that we think is happening that leads to the disease AMD? Well, here's a little bit of a cartoon, and I hope this is playing well on your screens, but essentially we think that light, chronic light buildup over time, causes your photoreceptors, and it doesn't matter whether or not it's cones or it's rods or cone dominant or rod dominant, the same mechanism happens. That causes them to be flooded with light. As that light builds up over time, it causes an increase in energy demands. It causes something called lipid peroxidation, which is the oxidation of lipids, and the buildup of reactive oxygen species. Now, these reactive oxygen species are really nasty. They can get into your DNA and they can start causing the cells to start to die. As these cells start to die, it causes a recruitment of your inflammatory cells called microglia, or recruited macrophages, into this space to come in and deal with this debris. But as a consequence of being in this environment, they get miscues and missignals, and they start just indiscriminately killing other types of healthy neurons, glias, including photoreceptors and other cell types of the retina. And that leads to rapid degeneration. So AMD is an example of a complex disease, okay? This is where you have genetics, you have lifestyle, so eating bad food, drinking alcohol, smoking, we know increases your chances, your prevalence of getting this disease, and environment. And I use environment with blue light because I know that I'm speaking to individuals who are constantly on their phone, like myself, and constantly on laptops, which emit a blue light, and we know blue light is very dangerous to our photoreceptors, and we fully don't understand the long-term effects that that's going to have with the next generation of individuals. So all these factors contribute to disease progression and AMD. So complex diseases are hard. They're going to take a long time to treat. But we have seen massive advancements in neuroscience and massive advancements in the treatments of retinal degenerations. So there is a simple disease called Leber's congenital amaurosis. Now, Leber's affects 1 in 50,000 individual. It's a degeneration of a protein that's found in the retinal pigmented epithelium called RPE65, and it leads to photoreceptors, both cone and rod death. Right? But it's a simple disease. That one gene, not a complex number of genes, one gene could be corrected. And in the last about 10, 20 years, we've seen advances that are enabling us for the first time ever to go in and actually manipulate that DNA or change that defective DNA and replace it with something that is normal and can function positively. And so in some groundbreaking work, what's been done is they took viral vectors that could be injected into the eye with a functional RPE65, and they deliver it subretinally so that it can get into the retinal pigmented epithelial cells, replace the defective RPE with a functional retinal pigmented epithelium. And I've got a little video here to show you some of the preliminary evidence that this was going to be functional. This was done back in 2008, right, this video. So what we have here is we have a patient who is going through a maze. This is a very dimly lit area. The people with Leber's congenital amaurosis, light is dangerous to them because the more light that they see, the more phototransduction that goes on, and RPE65 is very important in allowing phototransduction to occur. So here you have this individual, you can see them walking around on the screen here, then they're bumping into some of the walls, they can't really see what's happening, and you'll notice that over time, they make a number of mistakes. All right, it's taking them quite a bit of time to actually navigate through this maze. And I'm just going through this a little bit faster for the interest of time. So it took them around 77 seconds to travel from the start of that maze to the end. Now I want you to appreciate that after an injection, a single injection into the eye of a viral vector with retinal pigmented epithelium, RPE65 gene added into that mix, delivered into the patient the difference in the ability to navigate through this maze. Now, this is the first ever approved gene therapy for treatment of any disease, and it was done in the eye. This was done by a group called Spark Therapeutics that's now been bought by Roche, a really large pharmaceutical company, and it's amazing science, it's incredible technology, but there is one catch and one problem. It costs $425,000 US for one single treatment for each eye, right? So the question remains, we have some of these technologies for treating really simple diseases. We know that they're effective to improve people's lives. How do we go from transitioning from these simple diseases into complex diseases, and how do we reduce some of these healthcare costs associated with them? Well, I think there's only one thing that we can do, and it comes back to something that Helen Keller was talking about. It's about having a vision, and it's about education. And we believe that the way to do this and the way to solve some of these complex problems is really to invest in people, invest in their education, and invest in their research. So on that, I'd like to really thank my wonderful, wonderful group for all the hard work that they do. Really, they are the reason that gets me up in the morning, and are constantly amazing individuals to work with. And of course, our wonderful funding agencies, who without them, we wouldn't be able to do the work that we do. And lastly, what I'd like to leave you with is, if you want any more information about the work we do, please visit Clear Vision Research and the Eccles Institute of Neuroscience website, and you can all help us make this image clear. Thank you so much for all your time, and I'm happy to answer any questions you might have.
Thank you so much, Riccardo, for what a wonderful talk, especially the video that you shared about how one single therapeutic injection can make such a big difference in people's quality of life. And it's amazing that the research we did is contributing towards such changes. So if you're happy to take questions, the first question is, what are the best ways to prevent slow AMD? By Dave.
Fantastic question, Dave, and thank you so much for the question. So really the best way to prevent a large number of neurodegenerative diseases is to live a really healthy lifestyle and reduce your exposure to oxidative stress. So we know that not getting a good night's sleep can increase your oxidative stress. We know that not eating the right foods can cause an increase in inflammation, especially in your central nervous system, that can lead to degeneration. So eating lots of antioxidants, eating lots of good oils, right, and living a healthy lifestyle is the first thing. Second thing is not exposing you to anything that can cause degeneration, right? So each of us are unique genetic individuals that have a unique genetic signature, but certain genetics are going to make you more predisposed to developing AMD. So let's imagine you are someone who's predisposed to that. Then on top of that, you are overweight, you drink lots, and you smoke lots. That's going to increase your chances even more. So the best thing you can actually do is live a very healthy lifestyle, turn your laptops and computer screens off at probably about 8 o'clock, and allow the natural light that occurs in daily life to be your guidance of when you should get up in the morning and go to bed at night.
Thank you, Rick. At this one, I'd also like to say MDF, who are supposed to be there at the live event, they have some resources on their website and some fun recipes for summer, winter, like every season, and what are good foods and antioxidant-rich foods you can eat that, you know, helps you prevent AMD. So there's a lot of great resources on the MDF website if people want to check out. And the other question is, is glaucoma also related with reactive oxygen species?
Oh, good question. Correlatively, yes. So would there be an effect of individual who has glaucoma? Would that lead to an increase in reactive oxygen species? Almost certainly. So glaucoma is the death of essentially ganglion cells. So if you remember the retinal schematic image I showed, you had photoreceptors, then you had bipolar cells, and then you had ganglion cells. What happens in that is the optic nerve head seems to become sort of tightened a little bit, and that reduces the oxygen flow to those particular cells, and that can cause them to die over time. So if they start to die, you are going to get an increase in oxidative stress. You're subsequently going to get an increase in inflammation, and that can cause downstream effects. So yes.
Beautiful. Lots of questions coming in. The next one is, would the gene therapy used for LCA be effective for other rod cone dystrophies, or is it specific to LCA?
Oh yeah, absolutely, absolutely. There is for simple diseases where you want to go in and you want to just replace a particular gene, viral-based gene therapies are going to become more and more common. The problem is that it takes a lot of time to rigorously study this. I know that Roche and Spark Therapeutics have a number of different genes that they're looking at, but treating these simple diseases using viral gene-based therapeutics, absolutely. And we're going to see more and more of them come, not just for vision sciences, but many other diseases, including cancers, and maybe eventually some complex diseases like AMD, Parkinson's, Alzheimer's as well.
Interesting, yeah. Next question is from Felix. Does AMD eventually affect downstream transmission in visual-related brain regions?
Yes, so if you get death of the photoreceptors, right, eventually what will happen is if you don't have those communication strategies happening, the second order and third order neurons will also start to struggle. So you will get downstream effects from that. There is some really exciting research though of looking into whether or not you could tap into other parts of that cascade, including using ganglion cells that have their own light receptors to reprogram them to actually allow you still to see light. So that's one aspect. And the other aspect is there is a group of cells called Müller cells, Müller being a glial type, and I believe Brian talked about glia in his presentation, which what you could do is these cells seem to act like a bit of a stem cell. So you could go in and potentially reprogram those Müller cells to slot in to become other neural types, allowing that cascade to actually be repaired.
Wonderful. There's a lot of therapeutics in the near future. Another question is there any substantiating evidence that supports blue light blocking glasses as a preventive measure for AMD?
Well, we don't know directly whether or not for AMD, so I can't answer that, but we definitely know that blocking blue light is protective. And if you have a look online, there are a bunch of companies that actually produce blue light filtering glasses that have sort of a yellow tinge to it. And if anyone has an iPhone or an Android phone, you'll notice that there's a night mode on that. Now, in that night mode, it's essentially a blue light filter. So we know that technology companies know that blue light is dangerous. They have made measures to stop that. Whether or not it directly contributes 100% to the disease manifestation, or there's underlying other genetic consequences, we don't know. So doing a study where you just blocked blue light and saw whether or not it had an effect specifically on AMD is obviously very complex. But what we do know is that if you stop your exposure to blue light, you decrease your oxidative stress load, which we know if you decrease your oxidative stress load, you have less of a chance of developing AMD.
Absolutely, that was such a good question, but as you mentioned, it's a complex disease, so there are so many factors contributing, so it's hard to keep track of everything.
Absolutely.
Maybe we could take at least one more question, Rick?
Sure.
So I might just merge two questions together, if that's all right. So Yo asked if there are any differences between genders who are more susceptible to getting AMD. And then William has asked, does AMD affect some populations more than others, given that some region in the world gets significantly more or less sun? So
yeah, really good question. Thank you so much for that. So yes, there are gender differences. It seems to be slightly more prevalent in females, and we don't actually understand the reasons why. And there are definitely other genetic reasons and different populations that are more susceptible to age-related macular degeneration. So the lighter the color of the skin, the more susceptible individuals actually are to AMD. Now, exposure to light is a really interesting one to test, and there has been some studies that have done that. And what I would say from the data is that it's very much inconclusive. And probably the reason for that is that if you have individuals who live at, say, higher altitudes, who are going to be exposed to a higher level of light radiation, they are going to have been grown up in that environment and have genetics that make them protective against that increase in altitude, that increase in light exposure. So the experiment you would want to do is take individuals who we know are susceptible and move them to an environment where they are going to be exposed to a higher level of environment. Now, I don't know about you, but I wouldn't sign up to that literature. I wouldn't sign up to that experiment. And so the way you've got to do it is try and use models that enable you to answer those questions. It's a really wonderful question. Thank you so much for that, Will.
That's great. Thank you so much, Rick, for such an amazing presentation. And thanks everyone for all your questions. And I'd like to remind you at this point that our website has virtual booths for all the talks that were, part of this morning's session. So John's group has a booth in the website, Brian's lab as well, and Rick's lab has a booth on the website as well. So please feel free to check out the virtual booths, and you'll probably get more information about the research each of their labs do. And if you have any questions, feel free to write to them. This is the last talk for the morning session. We'll be back soon in 30 minutes for some more exciting talks about neurodegenerative diseases. But in the meantime, Angela will probably like to share a few entries we got to all the competitions that we have on BrainTeaser website, and also how you can sign up to maybe go into the draw of winning some goodie bags.